What I am about to present today is formulation and evaluation of herbal tablets and hard capsules containing Urtica Dioica soft extract. The presentation contents are Introduction, Research, Importance and Aims, Global, Recent Researches, Materials and Methods, Results and Discussion, Conclusion and Recommendations, and in the end the references. I'd like to start with botanical drugs definition. As FDA and NIH define botanical drugs, they are prescriptions that contain compounds referred to chemical groups produced by plants. They are main source in the alternative medicine. Botanical drugs have many advantages like safety, efficacy, availability, and they are cheap. On the other hand, they have many disadvantages, like the lack of legislation, few clinical studies, and unknown interactions between them and food and other drugs. Statistics has shown, according to World Health Organization, that 80% of development country people depend on traditional medicinal herbs in their non-severe illnesses. At the end of the 20th century, developed countries have oriented to use plants in medications to make a drug available for everyone, and because the medicinal herbs are effective and safer than chemical synthesis drugs. In the global market, botanical drugs production costs $62 billion. In the Syrian market, only 162 of botanical drugs are available. 42 of them are complementary supplements, but the problem is that the raw materials are completely imported. From here, the importance of the research arises to take advantage of the biodiversity of plants scattered in the Syrian environment by preparing pharmaceutical formulations containing cultivated extracts of these plants. So we aim to prepare ethanol extracts from Urtica dioica leaves to determine their total content of phenolic compounds and to do pharmaceutical formulation and evaluation of the prepared tablets and capsules containing Urtica dioica soft extract. Our chosen plant used parts are leaves and roots. The active compounds in the leaves are phenolic compounds, vitamins, and minerals, while in the roots they are polysaccharide, lignan, and glycan. According to World Health Organization, roots extract is mainly used in peniprostatic hyperplasia. This can be referred to its content of lignan and glycan, while the leaves extract is mainly used in joint pain and osteoporosis. This can be due to its content of phenolic compounds. Chemically, we all know that phenolic compounds are any compound which has in its chemical structure OH, but the botanical phenolic compounds, in addition to that, they must be syntheses from shikimic acid or polyacetate roots or both. These are a number of phenolic compounds in Urtica dioica leaves. Chemically, we all know that phenolic compounds are any compound that has in its structure OH, but the botanical phenolic compounds are, in addition to that, compounds which are syntheses by shikimic acid root or polyacetate root or both roots. These are some of the phenolic compounds in Urtica dioica leaves. Studies have shown that phenolic compounds lower TNF-alpha, they inhibit COX-1 and COX-2 enzymes. This results in prostaglandin synthesis inhibition. They also lower cytokines levels and lower CRP level. They also do scavenging free radicals. This results in antioxidant and anti-inflammatory efficacies for phenolic compounds. In 1990, a clinical study has shown that 50–80% of patients who took Urtica dioica leaf extract had completely cured, while 42% assured that Urtica dioica leaf extract was more effective than NSAIDs. In other studies, leaves have shown that they increase the therapeutic window of NSAIDs. As we can see that 50 mg of Urtica dioica leaf extract plus 50 mg of diclofenac, they are equal to the efficacy of 200 mg of diclofenac. In the global market, there are 1,087 products containing Urtica dioica leaves. According to NIH, 402 of them include their extracts. The official trade name of Urtica dioica leaves extract is Vitalgic. However, the leaves are traditionally used as tea or tinctures, but the recommended dosage of extracts are 360 to 600 milligrams, while the powder of the Urtica dioica leaves recommended dosage is 420 to 1000 milligram a day. In general, leaves of Urtica dioica are safe, but patients with hypertension or diabetes must be careful. These are some of the materials we used in this research. While methods included two steps, the first one, extract preparation, determined the best way of extraction of phenolic compounds, and the next step was pharmaceutical formulation and evaluation. For extract preparation, we followed these steps. But why using ethanol 95% as extracting solvent? It's because of its low toxicity and because it's good extracting solvent and because it's easy volatility. In this research, we compared three extracting methods. The differences were in temperature and time. We extracted at room temperature 40 Celsius degree and 70 Celsius degree, and for different time, 2, 5 and 9 hours for each temperature. The determination of total content of phenolic compounds were by using Folin-Cicalto reagent. The blue complex absorption was measured at 750 nanometer. The best extracting way was the way that gives the highest total content of phenolic compounds expressed in milligram of gallic acid to each 100 gram of the dried weight. This is the standard curve we had of gallic acid. The total content of phenolic compounds in mature leaves was the lowest when extracting for 2 hours at room temperature, and it was the highest when extracting for 5 hours at 70 Celsius degree. Same was for the young leaves. Extraction was the lowest when extracting for 2 hours at room temperature, and it was the highest when extracting for 5 hours at 70 Celsius degree. Comparing the past results, we came to that the best extracting method was by using young leaves at 70 Celsius degree for 5 hours. Our results agreed with past studies, as you can see. Our results can be explained that temperature makes phenolic compounds more soluble. This makes extracting for 2 hours at room temperature the lowest. On the other hand, temperature for a long time make oxidant. This makes extracting for 9 hours at 70 Celsius degree also low. For pharmaceutical formulation, as you can see that R1, R2, R3 has lactose as a filler, while B1 to C1 has calcium carbonate as a filler. B4 and C1 has magnesium stearate and sodium benzoate as lubricants. For formulation tests, we applied flowability and pressability tests by bulk density, tap density, Hausner ratio, and Carr's index. We also applied granules humidity test and the granules sizes distribution test. After formulation tests, we made appearance test, weight variation test, content variation range of phenolic compounds test, and disintegration time. We also applied friability and hardness tests as they are recommended by GMP. For granules humidity test, all formulations were in the recommended range for humidity, 2 to 6%. For density test, as we can see, only the formulation P4 was good and excellent as Hostner ratio and Carr index show. Formulations R1 to P2 were bad to weak, according to Hostner ratio and Carr index. B3 to C1, they were good as Hostner ratio show. This can be explained that the extract humidity was more than 7.5%. This makes calcium carbonate as a filler the best because of its good water absorption and because of its low water solubility. This makes a group which has lactose as a filler to be rejected. For lubricants, magnesium stearate, which was in B4 and C1 formulations, was the best because it's a hydrophobic lubricant. It's used with the plant extract in high percentage. Its hydrophobic polar coats the calcium carbonate molecules, and the hydrophilic polar is oriented to the pressure surface. Sodium benzoate was used in 5%. This was to decrease the hydrophobicity negative effect of magnesium stearate on tablet disintegration time. For other lubricants which were used in other formulations, they were hydrophilic and hydrophobic, but they were in low amounts. Therefore, only P4 and C1 formulations continued in the other tests. For P4 formulation granule size distribution test, we can see that the granule size were in the range of 500 to 849 nanometer. While for C1 formulation granule size distribution test, the granule size were in the range of 499 to 355 nanometer. For appearance test, we can see that the top list of P4 formulation were spherical with a smooth surface but non-homogenized colored. This leads that they need coating. While for the capsules, they were homogenized colored and the snaps are well sealed. And for the other recommended tests by the European Pharmacopoeia, we can see that B4 and C1 formulations were also in the recommended ranges. As I can conclude that we managed in this research to insert Urtica dioica leaves extract in pharmaceutical form other than pills, such as tablets and hard capsules. For recommendations, I recommend lyophilization for the extracts, and why not make a combination between Urtica dioica leaves extract plus vitamin D or diclofenac or zinc. On the other hand, why make pharmaceutical formulation for roots? Hope you enjoyed the presentation and thanks a lot for listening.